After necrosis and apoptosis, which were the more well-known forms of cell death, we are now going to start with the newer and the recent advances that have come up in the topic of cell death. The recent ones in the main exams tend to be asked more. So the first form of cell death, which is a more recent form that we are going to start the discussion with, is going to be necroptosis. Now, one very striking feature about this, you can see that this seems to be a combination. Necrosis, so that forms the first part, necrosis. And the last part is nothing but apoptosis. So what have they done? We have simply combined necrosis and apoptosis, and now we have necroptosis. Indirectly, I can say it will have some features of necrosis and some features of apoptosis. So remember, in terms of the morphology, it will be similar to necrosis. Was necrosis having inflammation? Yes. So this will also show presence of inflammation. So the morphology tends to be similar. Necrosis, as you remember, was showing cellular swelling. So yes, there will be cellular swelling. Then what does it get from apoptosis? From apoptosis, it gets a very important feature, that is the main mechanism. Now please recall, apoptosis was programmed cell death. This also, friends, necroptosis is also a programmed cell death. But then if both of them are programmed cell deaths, why have we made a separate term? What was apoptosis using? The main enzyme? Correct, caspases. So you can say that was caspase dependent programmed cell death. That is where the difference lies. Necroptosis is a caspase independent. It is a caspase independent programmed cell death. Over here, there is no activation of caspase. That is the main difference that is between apoptosis and necroptosis in terms of the mechanism. Now we come to the next part. There is one more feature of apoptosis that necroptosis shares. Remember, apoptosis could be both physiological and pathological. Same for necroptosis. Necroptosis can also be both physiological as well as pathological. So again, examples of these are important. When we talk about physiological necroptosis, the classical example that we need to know, necroptosis physiologically is seen when there is the growth plate development or the growth plate formation. So growth plate formation is an example of physiological necroptosis. When do we see necroptosis? Pathologically. Now pathologically, necroptosis is basically seen in a condition that is pancreatitis, inflammation of the pancreas. It is seen in steatohepatitis. Everyone is well aware, hepatitis, inflammation in the liver parenchyma. Steato means fatty. When there is fatty change also in the liver and inflammation, we will call it steatohepatitis. And the last example for this is reperfusion injury. Students, today we will discuss reperfusion injury because this is one of the very new and the important concepts that we have in cell injury. For the time being, you should know that pathological necroptosis is what is responsible for these three patterns. Now, just like we did the mechanism for apoptosis, same way we are going to go into the mechanism of necroptosis. So, what is the mechanism of necroptosis? Now, if you remember, I told you very similar terms of apoptosis and necroptosis in terms of the mechanism. So what are those similarities? If you remember, in apoptosis, we started the pathway with CD95. Yes. Over here, we are going to start the pathway similarly between two cells. But which receptors will come into play? there is binding of TNF receptor and TNF alpha. When there is binding of TNF receptor and TNF alpha, in that scenario, there is first recruitment of a complex. And what is this complex? There are three things that are going to come together. These include RIP1, RIP3, and please focus procaspase 8. Now this is a complex that is formed. When binding of TNF and TNF receptor takes place, there is a complex of three things formed: RIP1, RIP3 and procaspase 8. Now if you can just remember, few seconds ago we discussed necroptosis is caspase independent programmed cell death, and now we are saying that there is a caspase that is forming a complex. Please note I have used the term procaspase. This procaspase is only for the formation of this complex. Will it get activated? No. If this gets activated, which form of cell death will occur? Apoptosis. Procaspase 8 will always remain in the inactive stage. As soon as this complex is formed, these three things are put together, they are formed, there are two changes that take place. First, there is a decrease in ATP. The main power of the cell, energy of the cell, decrease in ATP. Second, there is going to be free radical formation. There is an increase in the free radical. Both things, as you can now understand, both the things are going against survival. ATP decrease, energy going down. Free radical increase means injury going up. In short, we are moving towards that these two things together will result in cell death. This is the mechanism of necroptosis, right? So now necroptosis, if you want to summarize, morphologically necrosis, mechanistically apoptosis, but caspase independent. It can be physiological and it can be pathological. Most important example for physiological, growth plate formation. Talking about the mechanism, the question that can come is, what is the complex that is formed in necroptosis? Correct. It is a complex that is RIP1, 3 and procaspase 8, finally resulting in cell death. So this was one of the recent forms of cell death that we have now discussed. On the same lines, there is another recent advance that has come, that is another type of cell death that we will talk about, and that is the next type, that is pyroptosis. That is pyroptosis. Again, a recent advance. Please remember, recent advances are very important. Pyroptosis is also a cell death, but pyroptosis is a cell death that is usually seen with microbial infections like Salmonella, Shigella. So the first point you need to know is that this is seen associated with microbial infections. Very important examples of these being Salmonella and Shigella. Now what do we need to know? For example, if this is a microbe that has entered the body, on the cell there is a receptor. In the cells there is a receptor known as the nod-like receptor. So the components of Salmonella, Shigella or any microbial species will bind to these nod-like receptor and will activate something known as the, very important information, it is the inflammasome. It will activate the inflammasome. Now, inflammasome is actually going to comprise of caspases. Which numbers over here? 1 and 11. 1 and 11. What is the main function over here? Remember, these are not procaspases, they are active caspases. So you can say that pyroptosis has active caspases. Caspase 1, caspase 11. Now these, that is caspase 1, is finally going to activate a very important step, interleukin 1. Now in our detailed discussion on inflammation, we will read that interleukin 1 is one of those most common cytokines which cause fever. Everyone knows that whenever there is any bacterial infection, person lands up with fever. Fever because of interleukin 1. Interleukin 1 getting activated because of caspase 1. So this activity of interleukin 1 will then result in fever. So this then formulates the mechanism for pyroptosis. So to revise, pyroptosis will be in response to microbial infections. What receptor will the microbe bind to? Nod-like receptor. Which caspases are activated? Caspase 1 and 11. Which interleukin is in turn activated? Interleukin 1. Caspase 1, interleukin 1. Finally, patient lands up with fever. So till now, we have discussed 4 types of cell deaths, and some of these had involvement of caspases. So I want to quickly revise those numbers because these numbers can in turn cause confusion. First caspase dependent programmed cell death was apoptosis. In apoptosis, the caspases for the initiation pathway were 8, 9 and 10, out of which 8 is not present in humans for apoptosis. Next, we had the execution pathway. Execution pathway had caspases 3, 6 and 7. 3 out of this was the most important. The next cell death that we did was necroptosis. Necroptosis was caspase independent programmed cell death, but which procaspase was present in it? Procaspase 8. Will it ever undergo activation? Not in necroptosis. And lastly, we did pyroptosis. Which were the 2 caspases for pyroptosis? Caspase 1 and caspase 11. So this is a summary of these 4 cell deaths that we did. Now moving on to another form of cell death, again an important one, the 5th form of cell death that we will talk about, and that is known as free radical injury. That is known as free radical injury. Okay students, this is also one of those topics which not only tends to be a favorite in pathology, but also in certain other subjects the same concept is going to get repeated because free radicals are formed all over the body. They will cause a damage all over the body and the entire body will try to fight against it. So this can come up in various subjects under various organs. So let's try to make a foundation and a basic concrete concept of this particular topic. First of all, what do we understand by free radical? Free radical is nothing but also known as ROS, that is reactive oxygen species. These are nothing but, if you go into slightly more detailed chemistry, which I am sure all of you remember, they have an unpaired electron in the outer orbit. That is what we had studied right in the 11th and 12th standard. They have an unpaired electron in the outer orbit. That is a free radical. That is a reactive oxygen species. They are in turn going to result in cell injury. So how are we going to study this topic? Let's see how they are formed, what damage can they do, and how can we protect ourselves against it. So first, let's talk about the formation of these free radicals. So let's consider this to be a particular cell. Let's consider this to be a particular cell. Now let's see, focus on the term. The term was reactive oxygen species. What will they form from? They have the word oxygen. So oxygen is going to get converted into something known as superoxide. How? This will happen in response to either some toxins, poisons, chemicals, or something known as reperfusion injury. On a daily basis, we are exposed to a lot of chemicals. So on a daily basis, a lot of oxygen is getting converted into superoxide. This is happening on a daily basis, remember. This is what your body is fighting against every day. The first step that happens is formation of superoxide. Indirectly, I can say this is the first free radical formed. Following this, the next step is that this superoxide will then convert into H2O2. What is H2O2? It is hydrogen peroxide. It is hydrogen peroxide. Because superoxide is converted into H2O2, the enzyme that is involved is SOD. And what is SOD? It is superoxide dismutase. Superoxide dismutase. So superoxide going into hydrogen peroxide in the presence of an enzyme superoxide dismutase. Now this H2O2 is also a free radical. Further, this H2O2 will then convert into OH, that is hydroxyl ion. That is hydroxyl ion. Now friends, this is a very important step that we have over here. When H2O2 is converting into hydroxyl ion, there is one thing that is helping this conversion. What is it? It is iron. The question will not be as simple as iron helping it. They will ask you which form of iron gets converted into which form. So remember Fe2+, which all of us also know as the ferrous form, gets converted into Fe3+, which we know is the ferric form. So you can say that the ferrous form is converting into the ferric form, and when this conversion occurs, then H2O2 converts into hydroxyl ion. This is a very important step that we have, because this particular step, just focus on this particular step, H2O2 going into hydroxyl in the presence of iron, this has been given a fancy name known as the Fenton's reaction. This is known as the Fenton's reaction. Very important question. The similar reaction can also occur in the presence of copper. This can also be associated with copper. When with iron, we are calling it Fenton's reaction, but the same reaction can also occur in the presence of copper. Now the next thing that we need to know. There are three free radicals formed: superoxide, H2O2 or hydrogen peroxide, and hydroxyl. What is the effect that they will cause on our body? So all three of these, whether we talk about superoxide, H2O2 and hydroxyl, they are going to damage every aspect of the cell. For example, the cell membrane has lipids. They cause lipid peroxidation. After the cell membrane, inside the cell, we have proteins. How do they damage the proteins? They cause cross-linking of proteins. After that, inside the cell, we have the nucleus. In the nucleus, they are going to result in DNA damage. So students, every cell has three important aspects. One is the cell membrane that has phospholipids. Those phospholipids have undergone lipid peroxidation, damaged. After that, the cytoplasm has proteins. Those proteins are damaged because there is cross-linking. Lastly, we have the nucleus. The nucleus has DNA. That DNA is also damaged by these free radicals. Now, because these free radicals are damaging our body and cells in every aspect, your body will always try to protect you. How does your body protect you against free radical injury? So that is again, in this entire topic, protection against free radical injury is actually more favorite with the examiners. So the protection against free radical injury. This will be best understood if you just remember what we had just studied, that mechanism, that pathway. First of all of us know and we are always told that we should consume those food items which are rich in vitamins like A, vitamin C, vitamin E. Why are we told that? Because vitamin A, C and E are antioxidants. Whatever free radical injury has happened in our body, it will scavenge, it will protect us. So the first thing is vitamin A, C and E. Vitamin A, C and E are important free radical protection mechanisms in the diet. Now, wherever vitamin A, C and E are present, they are protecting you. But the question that you get, which is slightly a link with ophthalmology that we have, where in the body or where in the eye will vitamin A not act as an antioxidant? So remember, vitamin A, C and E are antioxidants everywhere. They are antioxidants, but in the vitreous humor of the eye, vitamin A will not act as an antioxidant. Vitamin A is not an antioxidant. This is high yield information. Linking it with another subject, we need to remember all three antioxidants. Vitamin A is also an antioxidant, but in the vitreous humor of the eye, it is not an antioxidant. How is the body protecting you apart from this? If you just remember, we did a Fenton's reaction. What was it using? Iron. How else can that reaction occur? If there is utilization of copper. So if iron and copper are freely moving in the body will keep on using it for Fenton's reaction. In turn, what our body does, it has certain transport proteins. So if iron and copper are bound to these transport proteins, will they be available for Fenton's reaction? No. So what are those transport proteins? So transport protein for iron is trans because of transport and ferrin because of iron. Transport protein of iron is transferrin. What is the transport protein of copper? It is ceruloplasmin. So these two are going to bind iron and copper respectively. No iron and copper is available for Fenton's reaction. We are protected. The third protective mechanism that we have against free radical injury is the enzymes. Now, for understanding the enzymes, it is essential that we go through and revise that pathway once again. Superoxide getting converted into H2O2, in turn getting converted into the hydroxyl ion. Now students, what we need to know, if we want to break H2O2, which enzyme can be used? We can use catalase. Catalase will then break it into water and oxygen. Harmless things for our body, water and oxygen. So catalase definitely becomes protective for our body. Correct. But H2O2 will be broken down only when H2O2 is present. What if superoxide is present? Catalase cannot act on superoxide. Superoxide first has to convert to H2O2, then catalase can protect us. So superoxide conversion into H2O2 will be, just now we discussed, by an enzyme known as superoxide dismutase. Now superoxide dismutase can either be present in the cytoplasm or it can be present in the mitochondria. Why do we need to know these two separately? Because the form present in the cytoplasm is the copper zinc superoxide dismutase. And the form present in the mitochondria is the manganese superoxide dismutase. Very simple to remember. C for cytoplasm. Which form? C for copper zinc. M for mitochondria. Which form? M for manganese superoxide dismutase. So that is how we will remember. So you can say superoxide dismutase is protective because it converted superoxide to H2O2. H2O2 will then be acted upon by catalase and get converted into harmless products. So two protective enzymes we have done. Now we are left with the last and the third protective enzyme that can help us in detoxifying both of these. That is known as glutathione peroxidase. Glutathione peroxidase can act on H2O2 also. It can act on hydroxyl ion also and will convert it into water. It is a harmless product, water. So glutathione peroxidase is very important over here. Very important free radical scavenger which is going to protect us against free radical injury. How does it do that? While it is converting H2O2 and hydroxyl into water, the reduced form of glutathione will get converted into the oxidized form of glutathione. So from reduced to oxidized. Very important, because if we take out a ratio of the reduced to the oxidized form of glutathione peroxidase in a person, we can get to know how much of free radical injury is happening. Because with more free radical injury, more of reduced glutathione peroxidase will convert into oxidized glutathione peroxidase. In turn, more of the hydrogen peroxide and hydroxyl ion will convert into a harmless product like water. So students, for the protective enzymes that we just studied, roughly we have 3. Superoxide dismutase, second is catalase, and third is glutathione peroxidase. Out of these, clinical correlation for SOD is the most important. If we are particularly asked which enzyme is going to protect us against free radical injury of the brain, out of all these, the most important enzyme which will protect the brain will be superoxide dismutase. Superoxide dismutase. The clinical correlation that you need to know, there are some patients who have SOD gene mutation. Now, if someone has superoxide D1, particularly SOD1 gene mutation, there is a very high chance that that person can suffer from a lower motor neuron disease. So, SOD1 gene is a very important free radical protector in the brain. Someone has a SOD1 gene mutation, that person can land up in a motor neuron disease. Everyone has heard of a condition called amyotrophic lateral sclerosis. Amyotrophic lateral sclerosis or ALS. That is actually a disease that is due to SOD1 gene mutation. So first clinical correlate, first MCQ with this particular concept, very important. SOD is the most important free radical enzyme protector in the brain. And if SOD1 gene is mutated, we will have ALS. The next question that has been asked, out of the superoxide, H2O2 and hydroxyl, which is the most potent free radical? Which is the most potent free radical? That is, this free radical will cause maximum injury. What will you mark the answer as? It is the hydroxyl ion. It is the hydroxyl ion. Out of these 3, all are potent, but most potent is the hydroxyl ion. So students, this was the next form of cell death that we discussed, that is free radical injury. Before we end this, right in the beginning, I want you to recall we had done something known as, we'll get back to that table, we had done something known as reperfusion injuries. Right here I told you that free radical formation can also happen because of reperfusion injury. So this is what we need to know next. What exactly, just a few lines about what is reperfusion injury. So we will discuss this now. The next topic that we have is reperfusion injury. Now again, as I told you, the beauty of the subject lies that the topics are self-explanatory. Half our work is over if we split the term and we get the meaning out of it. So how do we derive the meaning from here? Perfusion means adequate blood supply. Reperfusion means blood supply was cut off and again it has been restored. So what do you mean by re? It is restoration. This stands for restoration of the perfusion. So let's take a simple example. There is a person suffering from coronary artery disease. Myocardial infarction happens in such a person because of coronary artery disease. What is the usual protocol? Either the patient is given thrombolytic therapy to dissolve the clot, or the patient has some stent placement done, again to make sure that the coronary vessels are patent, the blood supply is restored. Howsoever the patient was managed and the blood supply was restored. But now the patient's condition further deteriorated. So the patient suffered from attack of myocardial infarction. He was treated by any of these methods, but on treatment his condition further worsened. Why? Because of something known as reperfusion injury. So this means on restoration of perfusion, the patient condition might worsen. And why is this happening? This does not happen always. This is happening because of the formation of free radicals. When there is reperfusion that takes place, restoration of blood supply that takes place, there are formation of free radicals. That is what we had just discussed, the superoxide, H2O2 and hydroxyl. So this is because of free radical formation. Very important. Another mechanism that has been said for the same reperfusion injury, it says that when the blood supply was cut off, when there was a stage of ischemia, an immunoglobulin got deposited on the organ. For example, ischemia in the myocardium or ischemia in the heart. IgM gets deposited during that time. Now what happens when the blood supply is restored? When there is restoration, this IgM that is already deposited there, when it comes in contact with that restored blood, restored perfusion, there is activation of complement. There is activation of complement. So the complement system will get activated, the person will land up with more injury. The person's condition is going to deteriorate. So reperfusion injury, two mechanisms. One related to free radical. Second related to IgM, because IgM will activate the complement system. So these were the main forms of cell death that you needed to know. The last and probably the shortest form of cell death that forms the last form of cell death is something known as anoikis. Now students, this is something that is not very well dealt with in many of the guidebooks. Anoikis is definitely a question that has come, so we must know a line or two about it. It is a form of cell death, but it is a very simple one-line explanation that you need to know over here. Whenever a cell is growing in its natural environment, everything is fine, it is surviving well. You take that cell out of its natural environment, the cell will die. This is as simple as this. So, for example, if these are few cells which are growing together, so they are needing each other for their survival. Now, out of these cells, if I take out any one cell and detach it, if I detach the cell, it will stop surviving. So, if the cell is removed from its natural environment, it is going to result in cell death. That is as simple as what is known as anoikis. This form of cell death is known as anoikis. So students, these were the various forms of cell deaths that you needed to know. Precisely necrosis, apoptosis, combination of both of these, that is necroptosis, a newer one that is pyroptosis. Then we discussed free radical injury, which is also seen in which type of injury? Reperfusion injury. And the last one that we discussed, the shortest one, was anoikis.